I'm a research associate at St. Anthony Falls Laboratory at the University of Minnesota. And this afternoon, I'm going to present a summary of a project we've been working on the last couple years that's designed to develop and test methodology for sonar mapping of zebra mussel distribution and zebra mussel habitat. The motivation behind this project is really the lack of sort of information at a large spatial scale, the lack of ability to collect information at a large spatial scale on zebra mussel habitat and zebra mussel distribution. So current methods such as scuba diving surveys are really limited in the amount of data that you can collect. You might get some very high resolution, high quality information about how many zebra mussels are there and what kinds of habitat are they inhabiting in terms of the types of substrate, so sand, gravel, rock, etc. But you're really limited by the time and effort it takes in order to conduct a scuba diving survey. Similarly, other methods such as plate samplers or veliger or larval sampling of zebra mussels might tell you whether they're reproducing zebra mussel populations, but it doesn't give you very much information about where the zebra mussels are in a water body and how they're distributed. And so this project was really motivated by being able to gather a lot of information about zebra mussel distribution and habitat in a very large spatial area, to be able to answer questions like, where are the zebra mussels, how many are there, and how do their populations change over time. So the team for this project is a collaboration between folks at the University of Minnesota, including myself, Chris Millerin, and Andy Riesgraf at St. Anthony Falls Laboratory, Naomi Blinick and Chris Rounds, who are part of our scuba diving team for this project, and an undergraduate student, Gracie Mertes, who allowed us to compare our ground truth methods using scuba diving versus video and photography imaging methods. It's also a collaboration between ourselves and Macalester College, Dan Hornbach, Kelly McGregor, and Mark Hovey. And Mark Hovey is also at the University of Minnesota. Sonar mapping experts from the USGS Grand Canyon Monitoring and Research Center, and consultants who are also experts in sonar mapping methodology, including Matt Kaplinski and Cam Bodine, who is also at Northern Arizona University. And I do want to acknowledge that the initial project vision and advice on the development of this project was really provided by Dan Buscombe, who used to work at USGS Grand Canyon Monitoring and Research Center and has since moved to a different position. When I talk about multi-beam sonar, not talking about the kinds of sonar that you typically have on your fishing boat. I'm really talking about a research grade sonar. So it's a very high accuracy, high spatial resolution research grade mapping tool. And this multi-beam sonar can typically collect two types of data. We can collect depth or the bathymetry of a water body, and we can collect acoustic backscatter information. So depth from a sonar is typically measured by the time it takes a sound wave to return to the sonar instrument. In the field, this is, and especially with these high accuracy systems, it's often corrected by information on things like water temperature, which can affect the speed of sound. But depth or bathymetry is really measured by the time it takes a sound wave to return to the sonar. On the other hand, backscatter is really, you can think of this as a measure of the way that sound is reflected by the bottom and received by the sonar, the measure of the strength of the return. And it's this backscatter information that can really tell us a lot of information about the characteristics of a lake, ocean, or riverbed. So if we get a really strong return, typically you can consider this to be something like a hard bottom or a rock. Or alternately, if we get a weaker return, this might be something more like a soft bottom substrate or a muddy type substrate. This combination of information, the depth and acoustic backscatter from a multi-beam sonar, can provide information about a lake or riverbed at very high spatial accuracy. And we can use, and researchers have been using this acoustic backscatter information combined with field bed observations to create a model that relates acoustic backscatter to the substrate and bottom classification. So for example, these are data that were collected in a riverine environment where you can see that there's this map of acoustic backscatter, some information about the substrate at key locations that provides ground truth information that's used to develop a model. And then that model is used to create a map of the types of substrate. So in this case, it's sand and gravel, and rock within this river system. As we go back to thinking about mussels, and in particular zebra mussels, the physical presence of zebra mussels, you can think of this as it can change the roughness or the characteristics of a lake or riverbed compared to the same substrate, sand, rock, or gravel, without the zebra mussels. And similarly, other benthic organisms such as native mussels may also change the bed roughness, but they do so in a different way than zebra mussels. A lot of the native mussels tend to be larger organisms, and they tend to be thicker shelled, and so they create a different roughness signature than zebra mussels. And so we started this project with the question of, can we use this information, the information that the physical presence of mussels changes the lake or riverbed, to help us to identify mussel-containing substrate using acoustic backscatter. And so the first phase of this project was laboratory experiments in a controlled environment where we varied the acoustic settings, basically the frequency and the pulse length of the sound wave that the sonar sent out. We did this in a controlled environment where we had a known substrate and known mussel density. In order to do a first round kind of proof of concept, can we tell the difference in the acoustic backscatter signal between mussel-containing substrate and substrate that does not contain mussels? That experimental setup, which was conducted in St. Anthony Falls Laboratory, looks something like this. We had a sound-absorbing tank, and yep, that is a tank that is lined with AstroTurf, but the idea is to minimize any reflections of sound off of the tank walls itself. We had different sediment placed within the bottom of this tank, and then we had zebra mussels at different densities. High zebra mussel density, low zebra mussel density, and then we also set up an experiment with native Free Ridge and Blema plicata mussels contained within the substrate. And so for each of these scenarios, we collected acoustic backscatter data by varying the frequency and the pulse length of the signal into 12 different combinations. And what we ended up with was something that looked like this. And I don't want you to focus on the details of this plot, but rather to see that there's a lot of data here. There's a lot of information. And what you're looking at is violin plots showing distribution of acoustic backscatter for bare sand and gravel, for sand and gravel with low zebra mussel, high zebra mussel, and native mussels contained within it. And so the big take home here is that there's a lot of information here, but no single combination of pulse length and frequency, no single acoustic setting combination really discriminated between all of these groups. Create a data-driven model where we combine all of this information together in order to and relate it to what we know about each substrate. So what type of sand, gravel, or mixture is it, and what is the relative mussel density, and then use that information to create a model that we can relate the combination of backscatter features to the classification. And so in the laboratory, we were able to successfully do this. We were able to discriminate between sediment that had just sand or just gravel, had native mussels, or high or low zebra mussel density. But one key point that became obvious from the laboratory experiments is that the accuracy of the classification improved with a greater number of surveys, or more data going into the model equaled greater accuracy of classification. In practice, what that means is that each combination of settings of pulse length and frequency would be a different survey, passing over the same area using the multibeam sonar with a different setting. And so the accuracy increased, but it comes at a cost of additional data collection required in the field. Okay, So the next phase of this project was to take what we learned from the laboratory experiments and go out to the field and do a field validation. And the goal of this field validation was really to develop data collection and classification methods that take into account field variability. In the field, we have a range of depths, we have a range of substrates, we have vegetation, we have other things that can make this classification more difficult. And so we went out to the field, set up a series of transects and really targeted different types of substrates, so sand, silt, rock and gravel, and different mussel densities, so high, medium, and low densities, and also areas with and without vegetation. And for phase two, what that looked like is a series of transects, 20-meter long transects, that we had a team of scuba divers survey for relative mussel coverage, percent sand, silt, gravel, and cobble substrate, and any additional notes like things like the percent of macrophyte coverage within those each quadrat. And the scuba divers surveyed these 20-meter long quadrat, which were then surveyed using the multibeam sonar with this 12 different times with the combinations of pulse length and frequency. And then those two pieces of information were related together. One thing I want to point out here is that one of the challenges in combining the diver survey and the sonar survey is really knowing accurately where you are in space, or being able to tie accurately the diver survey to the multibeam sonar survey. The resolution of the multibeam sonar survey was on the order of 5 to 10 centimeter pixel sizes. And so being off by a meter or more in the scuba survey could really change what ground truth data you were relating to what backscatter information. So what we did here in phase two, we actually, at the end of each transect line, we had an object that would be really visible in the multibeam sonar surveys, in this case, large metal trash cans that helped us geolocate the scuba survey in our multibeam sonar survey. Okay, so we had two field sites for phase two, the first being White Bear Lake. And we selected White Bear Lake because we really, it was a local water body which had a range of zebra mussel densities, but also a range of substrate types. We were able to get sand, gravel, cobble, and rock within this single water body. And then our second field site was the St. Croix River. And we selected the St. Croix River again because we had a range of substrate types, but in this case, we had a range of native mussel densities present in these different substrate types. So for each one of our transects, we not only collected data using the scuba diving team and the acoustic backscatter information from our multibeam sonar, we also used an underwater remotely operated vehicle, ROV, which was fondly named Roberta. And we used this to not only create a verification video of each of the different types of substrate, but then also to then go back and compare our ability to tell what kinds of substrate and zebra mussel coverage we had using video alone to what the scuba divers measured in their quadrats. So what you're looking at here are three different images from the ROV and three different types of substrate, as a mix of sand, gravel, and cobble, mostly sandy site, and a site that was silty, but with some large isolated rocks present. And as an additional validation in phase two, we also had our scuba diving team select a small number of quadrats and remove the zebra mussels from those quadrats. We can then use this as a way of comparing the acoustic backscatter that was collected with and without zebra mussels, but over the same substrate. Okay, so for phase two, what this looks like, so here's an example of the bathymetry that was collected using the multibeam sonar around a isolated point in White Bear Lake, overlain with two transects. And those two transects were two of the transects where we had the scuba divers collect ground truth data and collected multibeam sonar acoustic backscatter information. If we zoom in on those transects, what you can see, the dots are the ground truth information from the scuba diving team, compiled at 2 meter resolution. So the 1 meter transects were combined into 2 by 2 meter ground truth points. And so what you can see, one of the transects was classified by the scuba diving team as sandy substrate with very low zebra density, while the other transect was still sand, but had some areas of higher or medium density zebra mussels. And that when we combined our acoustic backscatter information with our substrate model, we were able to map those areas of higher mussel density. So phase 3, the goal of this third phase was really to help translate this research into practice. And so the first step of this was to map habitat and mussel distribution at a large spatial scale. We are now working on taking the codes, the processing codes that we've used and modified for this project, and compiling those with our procedures in order to publish those and make them open source and freely available. And we're compiling our guidance for field data collection. In addition, the sort of last part of what we're working on here with our data is really compiling the limitations and challenges of the methods. So quantifying the error involved with areas of things like vegetation and what kinds of densities we're able to detect using these methods. Okay, so here's again, a map of the northeast corner of White Bear Lake with the acoustic backscatter information on the left-hand side that's overlain by our ground truth points, and on the right-hand side, our classified habitat map in White Bear Lake. And then here's the similar map in the St. Croix River, but in this case, we have sand and cobble substrate with high, medium, and low native mussel density. And again, on the left is the acoustic backscatter map, and on the right is our classified map of substrate and mussel density based on our data-driven model in the St. Croix. So one of the big take-homes that we learned as we were working through phase 2 and phase 3 is that in order to create a data-driven model of classification, you really do need accurate ground truth data. And in this case, we really need at least 2 points for each class. So each combination of substrate and mussel density, we need at least 2 points, one for training the model and one for testing the model. As I said, in acknowledging that scuba diving surveys can be time intensive and expensive, we wanted to compare what we were able to get using our scuba diving surveys to data that we collected using our underwater ROV and or photographs collected at each of the ground truth locations. And this exercise, while I think there are ways to account for these challenges, this exercise really highlighted some of the challenges. So again, I had a student here take data from the phase 2 field data validation and train on those data sets. So train on comparing the scuba diver data to the video data, and then take that training and apply it to the phase 3 or the ground truth locations for this third phase, the large scale mapping. And a couple things that we noticed right away, one of which is that generally speaking, using the video or photographs alone, the mussel coverage was underrepresented in areas with mixed gravel or mixed substrate, particularly with gravel. And one of the reasons for that, as you can see in the video that's shown here, one of the reasons for that is that it's the smaller live zebra mussels look very similar on a video to gravel pieces, and it's really hard to differentiate the two. You can see in this image here that the dead mussel shells are showing up very clearly, but the live mussels that are smaller and darker in color are very hard to distinguish between gravel pieces and mussels. The next thing that we noticed is that in areas where there's vegetation, again, using the video alone, the mussel coverage was really overrepresented in areas of vegetation. And that's in part because of our inability to see the actual bed or the actual substrate where there is vegetation. And similarly, we were unable to accurately classify the substrate using video alone in vegetated areas. And then the final point to make using video alone for either mapping zebra mussel distribution or for using as ground truth information in really turbid waters with low visibility. It's just difficult to see a large area of the bed using imaging alone. So one last thing that I wanted to point out here is again acknowledging that we've worked on developing these methods using what's considered a research grade instrument that requires not only an expensive piece of instrumentation for the Multibeam use it, acknowledging that while we were out collecting data using the Multibeam sonar and collecting all of this ground truth information, we also surveyed the same areas using an off-the-shelf side scan sonar system. So in this case, a Humminbird Helix 10. And so as a supplementary project related to our Multibeam sonar project, we're using this data that's collected from this off-the-shelf side scan sonar or recreation grade sonar to also see how close we can get in terms of classifying the information gathered by this instrumentation into either habitat maps or mussel distribution maps. Just a side scan sonar data, it's not really the same measure of acoustic backscatter, but we still get some information on the intensity of the return, which is still related to the sort of general substrate characteristics. So it's a similar type of idea. So again, we're working with a researcher, Cam Bodine, who's been working on machine learning models to map substrate types and habitat types using recreation grade sonar. He's been working primarily on models in the riverine systems in Mississippi, but we're now evaluating these models in White Bear Lake and the St. Croix River to see how well we can use these models to generalize into new systems. One of the big things that Cam has been able to do is create workflows to correct the sonar intensity in order to account for things that are create unrealistic characteristics. So for example, when passes overpass. So when the boat passes overlay each other. So if you look at this image on the right, you can see a lot of artifacts in the signal, and he's been able to create some correction workflows in order to clean that signal up and result in this figure on the left, which is our side scan map from the St. Croix River. And his initial results from the St. Croix and from White Bear Lake show that the model at this point has been somewhat successful at delineating things like patches of vegetation, sand gravel mix, and silty areas. But again, we're still working on this investigation into the accuracy of the classification. So taking a step back, so the lessons learned for applying sonar mapping to zebra mussel habitat. First of all, the mapping efforts at a large scale, they do show some promise. We're still working on this quantifying error analysis of the multi-beam versus the recreational grade survey, but we are able to get what look like very reasonable maps of substrate and mussel density. Or one point that I want to make is that as equipment improves, the cost and effort of the surveys using the multi-beam sonar equipment should decrease because newer equipment can cycle through some of these settings on the fly rather than doing individual surveys for each frequency and pulse length combination. We went into this project knowing that vegetation would be a challenge, and vegetation was a challenge. And it was a challenge not only in the survey information, but also in the collection of the ground truth data. It made it difficult to check our ground truth data using video imaging. Within the substrate mapping, the vegetation was a challenge, but we were also able, using in the multi-beam sonar data sets, to remove a lot of the vegetation within the post-processing. And the final lesson learned is that the accurate ground truth data is really critical to getting an accurate map. And this includes not just the information about what the ground truth point is, but also an accurate measure of the spatial location. And this is really important when relating the ground truth information to sonar information in areas of patchy habitat. But what this all means for zebra mussel management is that we can vastly expand the spatial area of current survey methods. We can make monitoring efforts more efficient by targeting areas of key habitat or targeting areas of likely high mussel coverage. We can also use these kind of spatial tools to help target management activities to areas where we have high mussel coverage. These methods could be used to map mussel populations over time and provide more detailed information on habitat. And as I said, the final code and the final error analysis is really due to be completed and distributed within the next few months. So with that, I want to say thank you. Thank you for dealing with some of the technical difficulties, and I'm happy to answer any questions.
We'll do this one first, Jess. Someone was wondering about the, you've noted a couple times in here very nobly, I might add, that you're going to include all the code online so that this can be open source and what you've learned will be shared. What are the sort of requirements there that you anticipate in terms of coding skill, in terms of processing power, in terms of space for data sets?
Mhm.
What kind of equipment or needs will you need to be able to run this kind of analysis?
So again, I'm recognizing right away that the use of the multi-beam sonar is a research grade equipment and it's research grade effort. That for the area of White Bear Lake that we surveyed, we have at 10 centimeter resolution. So what we end up with is a really large data set. So bathymetry and acoustic backscatter. And so we've ended up doing some of that analysis, some of that modeling, doing that using the Supercomputing Institute at the University of Minnesota. So that being said, I don't, one of the things that we're trying to really pin down as we're publishing these results is what spatial scale is needed. And it's likely that we don't need quite such high resolution data, which would make the data handling piece a little bit more straightforward. All of the code, all of the scripting is run in Python, by the way. So it's open source, but it's written in Python. For the side scan sonar, it's a lot less data, it's a lot lower resolution, and it has a lot less data computing requirements.
Thank you. I think this is a related question that just came in after this, so this may repeat some of what you've just said, but someone was basically wondering, is multi-beam sonar something that local managers could do with appropriate equipment or training, or is it just a research tool? And I guess you can answer that both in the now and also maybe in the future, or you can say, as technology improves, what might be possible. And they were also wondering, is this something you can envision somebody offering as a service at some point, or does somebody already offer it as a service, if you know of anything like that?
At this point, it requires a significant amount of expertise in order to run and post process the data for the multi-beam sonar. And so that really is something by those that own the equipment and have the post processing knowledge. Again, this equipment, it's changing all the time and things are getting a little bit more accessible, a little bit more straightforward. And I think there could be a point in the future where that is a more accessible option. Again, the idea behind looking also at the off-the-shelf, making maybe a less high resolution, maybe a slightly less accurate type of mapping, but doing so in a more accessible way. I think that there are examples for things like vegetation mapping where those tools already exist and are readily available. So
cool. Thank you, Jess. The next question I have here has to do with sort of resolution. I think it was a question of like, okay, so you're looking at an area, maybe your scuba divers are looking at a quadrat or something like that. And it's maybe 50% say sand and then 50% stone or gravel, like at a certain point your methods are going to have to characterize that spot as one substrate, correct? Or is there some way of like weighting or prorating how much of each sort of cell is actually made up of each substrate? Well, in other words, sort of what's the like resolution of these methods for being able to characterize substrate?
So that's a great question, and I a little not I didn't skip over things. I didn't include in for the phase 2 we actually did a examination of the sort of representing the zebra mussel coverage, and as the spatial scale increases, the effect of the patchiness kind of decreases a little bit. And so here's an individual mussel. So really we were targeting areas that were on the order of our ground truth information was 1 meter by 1 meter was the smallest resolution we thought that we could handle. But it's absolutely true that there's variation as you look at different resolutions. The other thing is that we did have to group our ground truth data into groups of similar types of substrate. So in order to create the model, we needed to have at least 2 points of each class of substrate and mussel combination. So if we had a continuous variable there with like continuously varying mussel distribution, you wouldn't be able to create this model where you could have a testing and ground truth type scenario with different classifications.
Thank you for answering that question. We've got time for one more here, so I think this is the one I'll pick. Someone was just sort of asking you to unpack a little further the relationship between sort of resolution and accuracy and water clarity. Was it linear? Was it really that just if the water was very murky, it was bad, and otherwise it was fine? They were sort of specifically asking, like, if you went to a very clear lake that had water clarity down 20 feet, if you would expect really high resolution there compared to other places. Sort of what does that relationship look like?
I think that there's 2 different things there. When I was talking about water clarity, resolution, and accuracy, I was specifically talking about the videos and photograph ground truth information. So if we're using our ROV with video imaging and we don't have great water clarity, we really cannot see what's on the bed in order to develop accurate ground truth information. Sonar, on the other hand, is able, and assuming that even in turbid water, although the what's in the water column does affect the way that sound is backscattered throughout the water column. But in theory, we should be able to see the bed and still relate it to the ground truth information that is on the bed. By see, I mean collect information about the backscatter that integrates what's in the water column and what's on the bed. And as long as we can relate that to the ground truth information, we can still create a data classification model. That's one of the reasons for accounting for variability, like things like water clarity, that you really need site-specific ground truth information as opposed to sort of a universal ground truth for substrate and mussel density.
Okay, thank you very
Did
much,
that help
Jess.
answer that question? I hope.
I hope so too. I will say too that I'm sure Jess would be happy to follow up with you via email if you would like to discuss this further. Her contact information is on the slide. So if it didn't answer your question, I'm sure Jess would be happy to continue that conversation via email.